Welcome everybody. Thanks for attending our Chestnut Chat today. Today we're going to have student poster sessions and flash talks. So I really appreciate all the support you guys are showing up for our student research today. So I'm Sarah Fitzsimmons. I'm the Chief Conservation Officer with the American Chestnut Foundation, and I think this is what, session 42. Kendra, is that where we're at?
Somewhere around there. Sounds
Somewhere
right.
around there. Yeah. We're almost to 50. Kendra pointed out that next year, is it June? June of
I think so.
24.
If we go every month.
If we go every month, we'll hit 50. So we're hoping to do something cool for our 50th Chestnut Chat anniversary. If you guys have any ideas, feel free to throw that my way for what we should do on June 2024. In any event, as usual, the only people who can share their microphone and their screen is me, Kendra, and our Cassie, who is also with the Chestnut Foundation. And then our currently will be 5 presenters. So Uma, Jeffrey, Tyler, Sky. We're the only people that can share our screen and our voice. The rest of you, we can't see you, we can't hear you, no matter how hard you try. It's not going to happen. I see a couple people with their hands raised. Generally, we don't call on folks through these sessions. And today, in particular, because we have several presenters, we're on a bit of a time crunch, and Kendra is going to keep us on time. So for the rest of the housekeeping, if you have something to chat, please throw that in the chat. What I've thrown down there is the poster session. So 3 of our 5 presenters have opted to participate in the live poster session that will take place immediately following our Chestnut Chat. If you have a question, please type that in the Q&A. Each of our presenters have 10 minutes to present. They'll do 5 minutes of questions following, and then whatever time we have left over, we'll do live questions at that. In the meantime, we can do some typing there in the Q&A module. Please put questions in Q&A. It's a lot easier for us to keep up with it there than in the chat. I think that's it. Before we go, and just in case, I want to plug next month's Chestnut Chat. We've got Michael French from Green Forest's work. He's going to be talking about reclamation of minelands, especially with American chestnut. So if you have any interest in that, I highly recommend, or even if you don't, it'll be a really interesting session. I hope you guys will come and join us next Thursday. What is that? November 17th is when that one will be. With that, I'm going to turn it over to Kendra. She's going to host and moderate our session from here. So Kendra, take it away.
Hi. Well, thanks everyone. I appreciate you all showing up to support our student researchers this morning. I'm Kendra Collins. I'm usually behind the scenes doing the Q&A, so this is a little switcheroo for me. I'm our director of regional programs and science coordinator for New England, and also de facto poster session and student talk coordinator. It's a role I've been serving for over 10 years now. So I'm going to give you guys just a quick overview of our researcher and student engagement programs that this sort of session today is a part of. So I just have a quick PowerPoint to run through, just kind of give everyone an overview of kind of what the sessions today are kind of a part of in terms of our larger programming. And then we'll get started with student flash talks. So let me see if I can share my screen. All right, are we good?
Yes, can see.
All right, awesome. So if any of you have been to our fall in-person meetings, typically we have kind of, a public program, and it's sort of a research conference vibe. We have presenters that we bring in that can offer updates on different research projects, whether those be scientific or cultural, really anything that we think might be valuable and of interest to our membership. However, we're always thinking about ways that we can expand that. And one of the ways that we have kind of tried to expand those programs and make more voices heard within sort of the chestnut research realm is through a few different programs. And so I'm just going to run through those really quickly. Our first poster session, the poster session was kind of the first piece where we tried to, rather than reaching out and inviting people to speak, we put out a call and said, if you would like to speak, let us know. And so the poster session is a way that researchers can share their projects with our audience. So we did that for the first time in 2012. We actually had a pretty good response. We had 27 posters. They were crammed into a very tiny function room. And I remember one of our founding board members, who has a somewhat imposing presence, he's a rather stern man, demanded to know who was responsible for this crammed room full of posters and people. And, I tentatively was like, well, I did this. And he was like, well, this is fantastic. We need to do this every year. So, excuse me, it's cold season. I have children. So that was sort of the beginning of our poster session. We've done that just about every year since 2012. We've missed a few. COVID for sure was a reason to put that on hold for a couple of years. In 2012, we started thinking a little bit more. We realized a lot of the presenters for posters were students, not all of them, and the poster session is open to everyone. But we did have a lot of students coming to our meeting in particular to present posters, and we figured maybe we could capitalize on that and make it a little bit more valuable of an opportunity for them by offering the opportunity to present at one of our sessions, presenter sessions. So we started doing a flash talk session that kind of fit in with the regular program. So we'd have, speakers who might speak for 45 minutes, and we'd have a 45-minute session of just students running through those. Our first student flash talk session, we had 5 students in 1 hour, which was kind of fun and sort of similar to what we're doing here this morning. So just to talk a little bit more about the poster session. So again, that began in 2012. We've run it most years since. I think we missed 2016 for some reason, and I honestly don't remember why. It must have been a logistical issue. Our peak was 27 posters. On average, we get about 16, which has been, it's a really nice opportunity to share a wider breadth of research with our audience. Just to kind of give you a sense of how those break down, 2012, again, was our peak year, but we have had pretty good representation every year that we've offered the poster session. And we've been able to provide a platform for 143 posters since we started this 10 years ago, which for a small organization and a relatively small annual meeting, I'm feeling pretty good about. I also just wanted to give you guys an idea of the players, the people who show up, what institutions are represented at our poster session. Many of these are folks that have only been one time, but we have a few standouts that, anyone who has any lead author university or research institution that has been represented more than 5 times, I called out here. Clearly, SUNY ESF, where one of our presenters today, Maya, hails from, has done a lot of posters. We've had 23 of their posters included in our poster session. UT Chattanooga, where Uma hails from today, is second place. So it's been really great to see that some of our research partners have participated in this session on an ongoing basis. Today's poster session will directly follow Chestnut Chat, as Sarah mentioned. It's running from 1 to 2, and the students who are participating in that this year, I didn't get any non-students, so it's just students this year. You can find their posters on our website at this web link, and it's going to look like this. There'll be a picture of each poster and a title. And the way you interact with that, there's a read more link below. You click on that, it'll open up a special page for each poster. So you can view the full poster, you can download a PDF, you can read the abstract. But if you scroll to the bottom, there's also a commenting feature. And so from 1 to 2, the poster presenters will be available to monitor and respond to those comments. And so that's kind of how we're handling the virtual poster session. Typically a poster session, you would walk up to the poster and talk to the presenter and ask them questions. Can't really do that as easily in the virtual space. So this is how we're handling that. And I encourage you all after Chestnut Chat to zip over to the website, check out those posters. And if you have additional questions that we didn't get to here, you can ask those at least of those 3 presenters. One of the sort of expansions of the poster session has been student poster awards. And so we began this in 2015, kind of on a whim. One of our education committee members at the time was like, hey, we should do poster awards. And so we scrambled to get a rubric together the same day. I think we gave out gift cards and chocolate bars to the winners. It wasn't very well organized, But it was the start of something. And so in 2017, we formalized that with cash prizes, which I think are maybe more appreciated than gift cards and chocolate bars. And the posters are judged on a few different things. And so this is also what our judges today will be judging the student posters on. So the content of the poster, the appearance and clarity of the poster, how well the information on the poster is organized, and then how well the student presents orally, like sort of talks about their research. And so because we're in this virtual space, the Chestnut Chat today, the flash talk that the students are giving will kind of stand in for that oral presentation. Just to give you an idea of who all we have had as winners and give everyone kind of a quick highlight. Here's our list of awardees. We missed 2020 and 21 for obvious reasons. But you can see there's a few universities and a lot of these are partner organizations for us that show up pretty regularly on those poster awards. So that's been a pretty nice program to offer and a nice thing that students can tuck into their resume. The last sort of piece to discuss is what we're doing now, and that's student flash talks. And so we began these in 2013, and we've made them a pretty regular part of our annual meeting programming. Just about every year. We typically get anywhere between 3 and 6 presenters. I think we usually offer them free admission to the meeting as a kind of a carrot for putting themselves on the hot seat. And certainly they can tuck that onto a resume as well. But we've had 26 different student presenters. Only 2 of those have presented more than once. I think one of those is actually Andy Newhouse, who is with SUNY ESF. I think many of you know who Andy is. The other was actually Jeffrey Wall, who is a student at Cornell working on chestnuts in Turkey, of all places. So we saw him a few times earlier in his research. So the way that things are going to run today, this is my second to last slide, so we'll actually get going here in a minute, is that each presenter is going to have 15 minutes, 10 minutes for their presentation, 5 minutes for Q&A, and then we'll move on. If it seems like folks are running over their 10 minutes and it's not clear they're going to wrap up, I will jump in and remind them of that time, but hopefully we can keep things moving. If you have Q&A questions, put those in the Q&A module. If you can, if it's for a specific presenter, should be pretty clear if it's right after the presentation, but it wouldn't hurt to drop in the topic or the person's name. And then this is the order we're going to go in. We'll have Tyler up first from William Paterson University, Sky from Clemson, Uma from UT Chattanooga, Jeffrey from Shippensburg University, and then Maya from SUNY ESF. And again, directly following our chat today will be the poster session. Again, here's the link, and I hope you guys will meet us over there when you wrap up here. And that's all I had. So let me stop sharing. Boop. All right. And looks like Cassie put the poster session link in again, so you can find that in the chat. And I am going to stop talking and not take up anyone else's time and turn it right over to Tyler.
Okay, let me just share my screen. Can you see it?
Yes.
Good? Okay. All right, guys. I am happy to be here. I'll just jump right into it. So I'm going to preface by saying I don't think I added where our university is located. It's located in Passaic County, Wayne, New Jersey. And the title is Restoring American Chestnuts at WPU, presented by me, Tyler Fishman. There we go. So I'm going to do a little about us. So the Department of Environmental Science at William Paterson University has been making some efforts to establish American chestnut trees around campus. We began our efforts of restoration in 2020, cooperating with facilities of the school. And this has required the dedication of faculty and students. And we our department encourages us to get involved in students alike with chestnuts through talks, club events, and club fairs, just to work with these wonderful trees. And here, this image right here is some of our alumni with our first generation of chestnut trees. And on the right is a picture I'll get into more later So staying in the loop, students and faculty alike have been working with certain organizations to acquire and learn about the American chestnut. We've collaborated with members of Southern New York chapter of the ACF, American Chestnut Cooperators Foundation, and as SUNY ESF as well. Members from those groups have come to our university to give talks and presentations to students. Students and faculty, we attend Chestnut Chats to attain the most up-to-date information about American chestnuts. And we are actively planting American chestnuts on our grounds annually. So that is something we have been making. So I'll get into some of our propagation methods. This is something I've begun this year, early this year. So the way I do it is the chestnut seeds are stratified in a stagnant peat in the fridge over winter. And to give them a good start once they germinate, they're given a mix of perlite, worm castings, and peat soil in quarter-gallon pots. In early spring, they're germinated in a heated greenhouse under artificial HBS lighting. These young trees are nurtured into healthy saplings, and they're moved outside once they are hardened off. And here is the process that I photographed of germinating American chestnuts from seed to get a little sapling. And this is the most critical time in the tree's life when it's most sensitive. And it's very important that you keep fungi away. Bacteria is very prone to attacking these young trees. Dampening off is something you have to worry about. So this is probably the most critical point in this tree's life. It's very interesting to see how quickly it grows. It's about 3 weeks difference from seed to young sapling. And note how hairy the seeds are. That's a very diagnostic feature of American chestnut seeds. So, and if you're lucky, eventually you'll get young trees like these. There's 5 in this picture, hard to see, but these are young American chestnuts about 2 months old, ready to be hardened off. And once they are hardened off, they are planted on our campus. So we take certain things into consideration, such as a light intensity, microclimate, water availability, foot traffic. Very important to think about when we were planting. They're protected in metal cages to prevent deer browsing, as well as buck rub, which can damage the stems of older saplings. And so that is important for keeping things like blight away, which take advantage of breaks in the tree to infect the tree. So it takes a lot of maintenance during the growing season because, well, this year we got a lot of rain, so there's a lot of weeds, and the mulch needed to be replaced, and we needed to assess the physical health of the tree during, obviously that's important during the growing season. And right on the right is a picture of our plantings throughout campus. And this map, there's little markers to where we have these trees planted. And this is growing, so we're working with our facilities to find more places to plant more trees. So we're going public with our American chestnut plantings. We have been utilizing ArcGIS software to create a digital story map of our chestnut trees on campus. So this is a little sneak peek on the top of our story map. It's not public yet, but we're working on it. We hope to share the story of the American chestnut as well as what we're accomplishing on campus with these trees through the collaborative effort of students and staff. And I spent my summer taking 360 pictures of all our trees on campus. So if you go on Google Maps of our campus, and you'll see our trees in 360 view, so you can take yourself on a virtual tour of sorts. So we are working with our facilities to designate a space on our campus for an orchard of these trees. And this is that picture from before. It's a little grainy, so it's taken with a phone. But it's about a 15,000 square foot plot of land designated for D58 trees once they get approval. So this will serve as a critical hub of for chestnut conservation. We're going to continue to work with the ACF and SUNY ESF to support us through this process. And our goal is to back cross our D58s and our pure chestnuts on our campus to promote genetic diversity, which is, of course, critical in restoration. So right here on the left is our Department Chair of Environmental Science, Dr. Nicole Davi. Here's Adriana Del Grosso of the SUNY ESF, Here's Tim Termini of our Southern chapter of the American Chestnut Society, Carl Petty of facilities, and Kay Gardner. She was an alumni that helped a lot with our restoration efforts on campus. And Kevin Garvey of facilities, Mike De Silva, our instrument specialist, and here's me. So, of course, American chestnuts are extremely ecological important. Their seeds are large and they're full of fats and protein. The bloom time of American chestnuts is extremely unique in that they bloom usually after all the other hardwoods have bloomed. So it's a very important and critical pollinator plant. And these trees were huge, so they produce a huge mass of pollen and nectar for bees and other insects. And many organisms rely on this tree, not just for its pollen, but as a regular source of food for its seed mass. Obviously restoring this integral part of our ecosystem is important for biodiversity because all our insects and generally everything in our ecosystem has been in a gradual decline. And this bee right here is Andrina reni, and it is associated with chestnuts in the plants in the genus Castanea. And it's basically gone locally extinct anywhere the chestnut has what used to be. It's been locally extirpated, and it primarily is found around chinkapins. Castanea ozorgensis and Castanea plumilia is where you'll find these bees now, but they're extremely rare. So you won't find any anymore, unfortunately. And of course, wood ducks and turkeys would eat seeds, and as well as bears and raccoons and all other sorts of creatures. So, and we had a very successful year this year with all the rain we got. Nearly all the trees on campus have doubled in size since last year. 2 of our trees we planted to replace stock that had been lost over COVID, and all trees remain healthy and blight-free over COVID. And you can see how small these trees are, and they're actually starting to grow out of their cages now because they're getting so big. So we'll have to get some bigger cages. And we also had our first big surprise with our first male catkin happen this year. So we look forward to future flowering events, and yeah, and then that's it. So.
Thanks, Tyler. There's one question. Kendra, do you want to take that one, or do we have time?
Yeah, absolutely. We are good. We have 6 minutes. So Ron says, thanks for your talk. What and where is your seed source, and do you plant hybrid chestnuts?
So our seed source is through we previously got it from Tim Termini of the Southern chapter of the ACF. So I've been working with my job in Oakland where I work for Restore Native Plants, and we get seed through the American Chestnut Cooperators Foundation. They have an annual seed list, and you fill it out if you're in the organization, and they'll send you about 10, 15, it depends on the season how many seeds they'll give you, but yeah, that's where we get our seeds from. They're pure American chestnut seeds. So
okay. So I assume the second part of that question, do you plant hybrid chestnuts, would be
okay. So we don't, and I have my poster, I'll get into that more. So
okay. We had a little flurry of questions come in. I think we can fill our 5 minutes here. So how many weeks do you do the stratification?
So it's over winter. So usually I think it only takes 30 days to stratify the chestnut seed. They have to be exposed to lower temperatures for a little bit. But I usually do it 2 months or so, and germinate them in early March in the greenhouse where it's warm and they have artificial lighting. So that seems to do the trick.
Right. What is the age of the tree that flowered?
Only 3 years old, so they flower young.
They can be quite precocious. All right. Cynthia asks, any advice on keeping the seeds free of mold when in the refrigerator?
So last year, so December of 2022, I just did it in this dry peat. And peat is a natural fungicide, very low pH, and it doesn't have to be moist. So if you keep them in dry peat, that should be keep mold away, and you should have viable seed.
Okay. Let's see. What type of soil mix do you use growing the seedlings?
So it's a mix of perlite for aeration, worm castings for nitrogen and other micronutrients, maybe a little bit of vermiculite. It's a little better at holding water, excuse me, than perlite, and then peat soil. So your regular, Home Depot gardening Miracle Grow soil. So you mix that together, um, it'll sustain them for a couple of weeks. Of course, they're trees and they're young trees, so they're going to be heavy feeders. So it's best to get them in the ground as soon as possible. We'll have access to more nutrients, but yeah, that's what I use.
Okay. Well, they look happy, so seems that your method is working for you guys. Anonymous attendee asks, do you have to use sunscreen cloth on your cages to prevent sun scald on your young trees?
So that's where the hardening off comes into play. So I'll take them outside the greenhouse and kind of put them in an area with decent exposure to the sun, but not too much. And gradually over time, you kind of move them more into sunlight. So it's not necessary, but I mean, if you're just trying to get these trees out as quick as possible, I'd recommend it. But if you have time to slowly what's the word adapt these trees to harsher conditions, then you don't really need to use shade cloth.
Yeah, hardening off is definitely helpful. Frank asks Tyler, why do you expect cages to protect from blight?
So cages don't protect from blight. They protect from deer. And deer are in some cases worse than blight when you're trying to grow seedlings, because they'll just come along and eat your seedlings. But blight, these trees aren't blight resistant at all. Nobody has D58 trees, well, the general public yet. So they won't, but trees can get big enough to flower, and we can collect germplasm from these pure trees, even though they are pure. So these trees are unfortunately fated to die one day, but we can still use them.
Okay. John asks, what's your enrollment at William Paterson, and how engaged in the effort do the students seem to be in general?
So if you mean my enrollment, I'm guessing you mean my major. So I'm environmental sustainability.
I'm guessing he's wondering how many students are at your school.
Oh, in general, a lot. I don't know the exact. It's a medium-sized university, so well over 2,000 students attend there. So I would say engagement is it comes and goes. We have people that are really into this and gung ho about it, and others that could care less. But that's why we have club fairs and talks to get people interested in the American chestnut. So people get engaged. You have to prove to them why they should be interested in it, and we try to do that. So it's here and there, but we do have people that are interested in our efforts. So.
All right, I'm going to give you one more question, and then
Okay.
I'm going to ask maybe if you could, as a panelist, hop on to the Q&A. You might be able to type answers to some of these questions while other folks are presenting if you would like to. Or if you don't get your question answered, hop over to the poster session to follow. Our last question from Russell. Russell is on here every month, so I wanted to get his question in. Do you have problems with groundhogs?
We don't, because they're caged. But I'd imagine if they weren't, we would, because we have gardens, native wildflower gardens on campus, and the groundhogs will just mosey on up and munch on everything there. So we have to fence those as well. So if you fence them and leave them alone, they should be fine from anything that's smaller than a bug can't really bother them. So I guess they could dig under, but we haven't had that.
Awesome. All right. Well, Tyler, you are all set. Thank you for your presentation. If you want to stop sharing your screen, I think we have Sky up next.
Yes. It's good for everyone right now.
Yep,
Yes.
looks good.
Okay, cool. So thanks for everyone's coming. My name is Xinya Lu from the Clemson University at South Carolina, the Department of Genetics and Biochemistry. And at the current stage, my project is about the histology of adventitious root formation and phytohormone analysis of American chestnut cuttings. So the first question is what adventitious root is. So in plants, roots that form from any non-root tissue such as stems, leaves, even the buds are known as the adventitious root. So from this photo, compared to the tap roots, the AR doesn't group deep into the soil, and the number of roots developed at a single point. The formation of AR plays a very vital role in normal plant growth and response to the abiotic stresses like flooding, nutrition deprivation, and wooding. And here I list several examples of the type of AR. The first one is aquatic roots, because of the flooding, and air roots, which is help to anchor the plant firmly. And the second, and the last kind of AR is from the side of wooding, and this is a major method to induce adventitious roots in my following project. Vegetative propagation by some cuttings is most commonly used method to propagate many wood plants clonally, since it's easy to apply and usually result in high rooting percentage for many plants. Asexual propagation is small, energy efficient, and the offspring produced will share the characteristic of their parents identically, but the reason why some species are reconcentrated is still unknown, which includes American chestnut. And we just think the process of cutting also use hormones before plantation to induce root development. And the formation of AR is a complex developmental process and is highly regulated by hormone crosstalk. Therefore, the induction of AR is a bottleneck of the several of cuttings. So it's essential to understand the regulation of excision-induced AR formation during this whole process. So to answer this question, we made a comparison of hormone levels of American chestnut stem cuttings. The endogenous hormone profile cuttings is not in favor of AR induction, and from this graph, compared to the easy-to-root popular, the levels of known AR inhibiting hormones such as cytokinin, IASP, ABA, JA, OPDA, and SA were significantly higher in American chestnut. In contrast, the IA level was significantly lower in American chestnut compared to the popular. And in addition, the T-zerotin, C-zerotin, DHZ, and the TZR are biologically active forms of cytokinins. However, neither C-zerotin nor DHZ were detectable in the cuttings of popular and American chestnut, but T-zerotin was detected in American chestnut cuttings, but not in popular. As for TZR, its level was doubled in American chestnut. And as we know, the cytokinin signaling and perception are known to be necessary for root development. However, it is reported that the cytokinin shows the negative effect on the formation of adventitious root. And they also try to explain the formation of adventitious root from the histological analysis. And this stem structure was divided into pith, xylem, vascular cambium, phloem, sclerenchyma ring, cortex, and epidermis from inside to outside. The pith is located in the center of stem, and the xylem locates between the pith and the vascular cambium, which accounts for the large portion. And the vascular cambium was between the xylem and the phloem. And the sclerenchyma ring was annulus with a distinct staining. The cortex was mainly composed of loosely arranged cells. And last one, the epidermis is located on the outmost side of the stem, and the epidermal cells were closely arranged. So the continuous tissue section observation indicates that the root primordial might originate from the callus, xylem, phloem, or the vascular cambium. But at this stage, the process shows the differentiated cells produce various types of cells in the phloem, some of which differentiated slowly into in some areas, and in this region, the cluster of meristematic cells composed of differentiated mature phloem cells were large and had strong meristematic ability, processing the context to bulge outwards. But unfortunately, I haven't got the perfect photos to show the root primordial and the mature adventitious root from the American chestnut stems. But in our hypothesis, the AR formation phases were divided into 4 different stages. First one, induction, and at this stage, the cells become meristematic. And second one, the initiation phase contains cell division, formation of meristematic centers, and following by the expression. And at this stage, we can observe and found the emergence of root primordial. And the last one is the root elongation. And after that, we can find the mature adventitious root of American chestnut stems. And I think that's all. So have any questions? I think I can yeah.
All right. Well, thank you so much for that. You know, rooted cuttings is one of those things that we've always wanted to be able to do. It's a difficult nut to crack. So far, we've got one question from John. He wants to know, do trees grown from adventitious rooted cuttings form root collars?
Actually, some of them, just some of them, because adventitious root can be induced from the site of woundings directly, or we can induce the adventitious root from callus. And from the root columns, I just observed a small percentage of the examples in my as a current stage of my project. Did that make sense?
I think so.
Yeah.
Any other questions? Well, I'm gonna pull a couple from the chat, even though we would prefer folks get these in our Q&A. Let's see. Leslie just says thank you. She enjoyed your presentation. William wants to know, can a person use standard available root hormone from a lawn and garden center?
We use NAA mainly, and the concentration is 100 microgram per liter.
Yeah, and I'm honestly not sure what you would get from a garden center. I don't know. I see Hill on there. I don't know if he would know better. I guess compare what you can get at the garden center to what they're using in the lab. Let's see. Did your studies suggest any change in the technique of rooting cuttings?
Actually, no. We just use very traditional cuttings technique to induce the roots from the American chestnut.
Right. Let's see. Have you studied hormone levels in American versus other species of chestnut?
We just compared the hormone level between the American chestnut and popular, and we just found the significant difference between in the IA, and we think it maybe just the mainly maybe just the main reason that's why the American chestnut is difficult to induce rooting.
All right.
Mhm.
Thank you. Let's see. Where are you in your study and what are you doing next?
I'm from the Clemson University in South Carolina, and in the future, first I will finish the historical analysis of American chestnut and try to make up the gap between these four different stages. Yeah.
Great.
Mhm.
Let's see. We've got another question. Were the poplar cuttings you used Tulipifera lyrodendron? Do you know which species of poplar you were using?
I'm sorry, I have no idea. It just
That's okay. All right. Paul wants to know if you have grown roots on chestnut cuttings.
Yeah, of course. We have we already finished the establishment of the mature roots, and it looks I didn't put the photo in my slides, but yeah, we already got them.
Great. Well, that's exciting stuff. Let's see, what else do we have here? We've got a couple more minutes. Oh goodness, a lot of them. Oh, Haiying said it is populous for
Haiying Liang is my advisor. Yeah.
Thank you, Haiying.
She's here. Yeah.
All right, we got that. Let's see. Is the root quality about the same as the poplar cuttings?
Actually, they're totally different, because for the popular cuttings, and we can induce rooting from the popular within just 1 week. But for the root quality, I have to say the root quality of popular is more healthy than the American chestnut, because generally speaking, for my results, the American chestnut can be induced just a limited number of roots, but popular, they're very good. Yeah.
Yeah. Yeah, I feel like they're two very different beasts when it comes to trying to get them to root. Fred asks, are you experimenting with applying hormones in different combinations to induce root formation?
Yeah, but it's because of the limitation of funding and the time. Yeah, I were considering about the different combinations of hormones, but it's just in plan. Yeah.
Yeah. Understand.
Thank you.
That time piece is often a limiting factor, but I know your advisor Haiying has been working on this for a little while, so I suspect that work may continue. John asks, I guess mature, oops, something moved here on me. Let me get back to John's question. I guess mature is a relative term, but do mature induced roots show any histological differences from naturally formed lateral roots at comparable sizes?
I'm still working on it, because I don't have at the current stage, I don't have the perfect photos to show the stems with the mature adventitious root. So maybe I hope I can finish the historical analysis by the end of this year, and we will find the difference. Yeah.
Well, maybe we'll just have to have you come back and and share an update.
Yeah.
All right. Leslie wants to know if you know if chestnuts can be propagated with air layering.
The air layering?
I suspect not, but
I don't think so. I'm sorry. Yeah, it's
chestnut is
too
pretty
hard.
tough.
Yeah.
All right. Let's see. We have 30 seconds. What age were your stock or parent plants? Did or will you study the age effect?
You mean the age?
The age of your stock or parent
Yeah,
plant?
yeah. I were considering the effect of the age, but the American Chestnut carton is provided by the Chestnut Foundations, and I have no idea about how many ages. Sorry about that. Yeah.
Fair. All right, my timer is going off. We are at 15 minutes. So Sky, you are off the hot seat, but thank you very much. That was a great presentation. And I think up next we have Uma.
Hello. Okay, can you guys see that?
Yep, we can see your poster.
Okay, perfect. Hello, as Kendra said, my name is Uma Plambeck. I'm an undergraduate at the University of Tennessee at Chattanooga. I'm studying environmental science, and I'm doing my thesis project with Dr. Craddock, who you've seen floating around the screen. And I'm doing my project on screening full sibling and half sibling families of chestnut seedlings for their resistance to oxalic acid and Cryphonectria parasitica. And this is the poster that will be on the website later, but I wanted to blow up some parts of it. So this list is the families that we used throughout both projects. It includes 8 species. So we've got C. alabamensis and ozarkensis, which are the chinkapins. And then we've got dentata, which is the North American chestnut. We've got crenata from Japan. Millissima, Cygwinii, and Henryi are all from China. And then sativa, which is from Europe. And then we also have the 9 families here, which are the from the 2022 Best by Best crosses, a lot of which are from the Tennessee chapter of the breeding program. And the seeds of all the species and the hybrids were planted into 5-liter containers in late February, or in late winter in February. And they were planted by the UTC greenhouse crew, which is run by Dr. Craddock, and we have 2 paid interns and several student volunteers. And when the weather settled, we brought the seedlings from inside the greenhouse outside onto the drip irrigation system and divided them into 10 completely randomized blocks. So one of the projects that I was doing was oxalic acid, an oxalic acid assay. And so I ran a trial to determine if leaf position on the tree matters, or if it's statistically significant, and it was. And so I then ran another trial, and I tried to sample every single tree, and I had 967 trees. So 1 leaf from 967 trees. And that was too many leaves for me. And so I tried it again. And for the 3rd trial, I collected leaves from 2 blocks that were pretty far apart from each other to get a full like setup of the greenhouse. And so I sampled about 200 trees, and I took 3 leaves from each tree that were right next to each other. And I cut out 15mm discs, which is what you see in this picture. And I cut about 3 to 4 leaves, or 3 to 4 discs from each leaf. So 10 discs per tree. And I set them up like this. And then I took, or I soaked them in oxalic acid in 50mm or micromolar, sorry, oxalic acid. And then they were rinsed and dried. And then I took photos of them, which is this picture right here. And you can see the browning, which is the reaction that I was looking for. And the photographs were analyzed using image J and R. So I measured the browning reaction and then analyzed it. And then for my small stem assay, I inoculated the plants with a very large team of 15 people on July 17th, when it was very hot and very humid, and took us a lot of water and a very long time. to get through all of the trees. And then 12 weeks later, I measured the cankers and the orange zone on October 9th, as per the Cipollini et al. Methods. And then I analyzed that data using R. So for my results, this is the browning of oxalic, or of leaves in oxalic acid, but I'm going to talk about the cankers first. I compared the full canker length by family so far, which is the figure that one of the figures that you'll see on my graph. And I would like to admit that this is my first try on this experiment, and so it's not a very exciting graph, and I'm not very satisfied with it. So I'm going to go through and attempt to fix it and measure or analyze the orange zone instead of the full canker length. But I had a few questions about some trees and their orange zones or lack of orange zones, and I wanted to investigate that further. So better results to come. Now onto the figure that you see. You can see that the oxalic acid shows a much larger difference between families and potential resistance to oxalic acid. So this third box plot here is the C dentata, and then the C mollissimos are these two, and they differ based on whether or not they're from Alabama or from Georgia. So this is the Alabama group, and this is the Georgia group. And then you can also see all of the test crosses right here. And those also vary greatly in their resistance to oxalic acid, which is comparable to either the dentata or the mollissima based on which family you're looking at. And then this is me. Dr. Craddock made me pose for this picture. But for further investigation, I plan on testing variation in oxalic acid and Cryphonectria parasitica response for statistical significance. And I also plan to run the correlation between oxalic acid resistance and C. parasitica in R. And I'm going to run a regression of the orange zone versus the full canker length, so I can make the decision on which is more statistically significant, and create another graph with the orange zones versus the families once I get my clarification on the orange zones. And I'm going to improve upon the figures that I already have and rank them by resistance instead of rank them alphabetically, so that the figures are easier to read. And I hope that the results of my thesis will contribute in some way to the restoration of the American chestnut. Thank you for your attention, and are there any questions?
All right, Uma, thank you. And I'm glad Hill made you pose for a picture. It's a very nice picture.
Thank you.
So, oh goodness, I just put my video in a weird place. Okay, so we have a couple questions coming in. We have a few minutes. First question, and I think this may just be a clarification. I'm curious about the Alabama and Missouri species. Do they still belong to the American chestnut family or not?
The Alabamensis is a chinkapin, and so is the Ozarkensis. So yes, they're part of the same family, but they have some slightly different, they're different species and have different characteristics.
Great. Yes, those are, I consider them cousins. I don't know.
Yeah, that sounds right.
All right. How does the browning of the leaves affect our understanding of these trees?
So the oxalic acid browning reaction is a relatively new system. And so what I'm trying to see is if it is going to produce the same results as the SSA.
All right.
Yeah.
Great. Any other questions? Let's see. Wow. All right. It always takes, it's a little lag for folks to start typing. Why do you use oxalic acid?
We think, based on other studies, that the oxalic acid is what the Cryphonectria parasitica uses to kill the trees.
All right. Back to, I think this is a follow-up on the chinkapin question. Are they similar in size to dentata?
Yes, their leaves are smaller. I think that's the biggest difference from what I've seen.
Right. Yeah, and these are all greenhouse grown plants
Yes.
at this point, right?
Yes.
Yeah. Yeah, I think Hill can correct me if I'm wrong, because I don't have a lot of chinkapin up here. Typically, in a perfect world, they don't get as big as dentata, but American chestnuts don't often make it to become big trees.
Yeah.
We think of chinkapin as being more of an understory species, but then they do weird things like hybridize and, I don't know, turn into the albumensis is sort of the pocket trees, right? So those actually look more like a dentata.
Yes.
And then I think Fred Paye did some interesting work with Ozark chinkapin and mapping kind of what those logs look like, and they were actually much more tree shaped than I think the literature had us believing. Let's see. I missed it. Are all the hybrids B3F3 generation?
No, it's just best by best.
Those are F2s.
So they're F2s.
Okay, awesome. Oh, for the chinkapins, there was confusion. Size of the nuts. Are the nuts the same size?
They're very small.
Yeah, I think one of the distinctions with chinkapins is there is one nut per burr, and they're like the size of, they're like a little grape, like the size of a
Very,
pinky.
very little.
All right. Well, Uma, I think you did a good job explaining your project. Because I think we're out of questions unless there's anything.
There's one more question in the Q&A.
Oh, great.
Or sorry, in the webinar chat, sorry.
Okay.
Do you include a pH control?
I have not yet, but that is a good idea, and I'll put that in for future investigations.
Awesome. It's only October.
It is only October. And again, I collected all of my data on the 9th, so very quick turnaround time.
Awesome. Well, thank you for doing that. All right. Well, great. Well, Uma, thank you so much.
Thank you.
I think we will just keep things moving, and we'll switch gears. We'll get away from the hard sciences quite so much, and we'll hear next from Jeffrey.
Alrighty. Hello, everybody. I am Jeffrey Grimes. I'm an undergraduate at Shippensburg University. My research advisor is Dr. Alan Dietrich Ward. He is a professor of history. He has a focus in environmental history. I got talking to him because of my interest in the American chestnut. And we got started on this last semester. I put in a proposal, and now my research topic is Castanea species in the Mid-Atlantic and Apennine Peninsula from Cryphonectria to contemporary. So the actual research that we're doing is asking the question, how has research on Castanea species in Italy and the United States transitioned from a primary focus on Cryphonectria parasitica to addressing the broader environmental challenges that conservation efforts face? Now, this is important because we're trying to find out how much organizations such as the American Chestnut Foundation and various groups trying to do research on chestnut species have shifted from looking at blight to various other issues, from diseases to pests, insects, fungi, what have you. Now, this topic is interesting to me because I come at this from an angle of comparative analysis. I'm looking at this as both a biological, environmental, and an international studies angle. And my background in social studies and history influenced this heavily. So I've only been looking at this for about 6 months, and this semester I've been able to look more into the literature, both the primary sources and secondary literature of books, and there's also been lots of journals I've looked into. And it's quite striking to me the difference between the American and the European, or specifically Italian, approaches of handling both blight and current problems that Castanea species face. Now, my research advisor and I, have been focusing in on the PA chapter, and specifically the Apennine Peninsula in Italy, because of the specific issues that they are looking at. So issues with reintroduction in the United States and Italy both have looked at chestnut blight, which is Cryphonectria parasitica, coming through the cracks in the bark and going to the cambium later. And then there's also been ink disease, which is Phytophthora, and it moves between trees, water stagnation, it goes through the roots. But there are also other issues today, which we look at, from brown nut rot to wasps and moths. And then there is various issues from not only in orchards, but also in the wild, which we see with chestnuts. All are considerations that the American Chestnut Foundation and various organizations trying to reintroduce American chestnut need to look at to be able to succeed. Now, the research institutions that I've been researching and looking at their historic focuses and aims and goals have been the American Chestnut Foundation, specifically the Pennsylvania, New Jersey chapter, which is the main case study of my historiography. Hopefully, I'm looking to talk with some of their members, the board members, and I aim to do an oral history. I've talked to several people, mostly via email, but I plan to talk some more with them. I've spoken with Dr. Marshall, who was a plant pathologist at Shippensburg, who used to work with the PA chapter, and he's retired now, but he knows quite a few members of the chapter. Then Penn State University is another organization, university, that I'm using as a case study. Their selective breeding program has been very important when looking at Castanea dentata and trying to reintroduce that. I've been looking at the records at the Paterno Petit Library in the Everly Family Special Collections, and that's where I've been doing the majority of my archival research, getting into the weeds of what actual sources have been collected and stored over time and how things have changed. Just the viewpoints and perception of both the public, the political, and everything that needs to be considered. So on the European side, the Piedmont Regional Chestnut Growing Center in Italy is, in conjunction with the University of Turin, does research, and they suggested to me the Chestnut Handbook, Crop and Forest Management, which is the aspect of the Italian side that I'll be focusing on, as well as the Chestnut Study and Documentation Center, who's also been doing research on Castanea sativa, excuse me. Now, the differences in approach that I've seen between North America and Europe, honing in on the United States and Italy. In the US, there's a big push for American chestnut is something that grows in the wild. It is natural. It is something that to be conserved, restored, and wild. In Europe, in most of the literature that I've seen and to the people I've talked to, it seems more commercialized. So Castanea dentata, more in nature. Castanea sativa is more for orchards. So they do high density orchards for nut production, whereas we are aiming for trying to replant Castanea dentata in the forest, the hardwood forests where it once was before the blight. Now, I'm looking at various reasons why that might be, from the date of when the blight first came into the countries, to political ideologies, to social and cultural importance of the trees, and various aspects of what could have caused this. So another aspect of this is, in the United States, there's a large push for American chestnut purity, and in Italy, they have various different mix and match. They have, as you can see here, 150 different grafted cultivars. They use sativa, millissima, dentata, pumila, granata, all different kinds of hybrids and genetic variability, all for the best nut, for the best tree, whereas we're focusing on trying to keep our nuts and our trees American. Now, while I'm doing this research, compiling resources and information, I try to keep some things in mind, looking at the broader implications of the shifts in focus, looking at the historical aspects as well as the context of what was happening at the time. The timeline of this starts from the early 20th century and moves into today. And the largest push, the shifts that we're seeing, it didn't happen overnight. These are changes that happen between different presidents, different time periods, and all of that is what I'll be considering, as well as the different problems. So introduced pathogens, introduced pests, they all come at different times, and these are things that will need to be looked at in relation to this research. So why the PA New Jersey chapter? Well, obviously Shippensburg is in Pennsylvania, but I'm also located on the northern part of the Appalachian Mountains, and this is where the American chestnut was and still is in some times. And here it was ecologically, economically, and socially important. And the research that the PA chapter has done is what interests me, as well as the records that they're able to keep. If anyone has any questions or would like to make any comments, just please let me know.
Well, as one of the hosts, I'm going to ask a question. While other people put that in there, I'm going to take that privilege. So I'm curious about your experience using the archives at the Paterno Library. Would you mind sharing just like kind of your experience going in there and finding things? And selfishly, because that was Stephen Hoy, who I think is on here, myself and a couple of volunteers, having zero archival education, we did our best to organize it. So I'm just curious about your experience and if you have any possible suggestions, because we intend on putting more in there too. So if you have any suggestions of what we could do to make that better, that would be great.
So one of the aspects of my research that I found a bit difficult is the records stop at 2014. So there's 15.6 linear feet of boxes. I've looked through about a fifth of that so far. I'm about 2 hours away from the library, so I've only made one trip so far. I've looked at a lot of stuff online, but it made me feel like a historian going into this special little room, and they plunked a big old box right in front of me. I took off the lid, and it's just manila folders full, like this thick, of just various documents. And I was looking through email chains between members. I was looking at flyers that they've sent out, information. There was everything from stickers to there was a whole two pages of basically important contacts of people who've worked on this incredible project. And seeing the amount of variation of information, the kinds of backgrounds of the people who work on it, the kinds of organizations who've helped, everything from people who've donated to people who've volunteered, all of that information was really helpful.
Nice. Well, I'll say, come back to the library, of course, but also you're welcome at our office to check out the 2015 to present archives as well as we put those together. So thanks for all that you do. Kendra, sorry. Thank you.
Sorry, just finding my unmute button. No, Sarah, I'm glad. I was very tickled to hear, Jeffrey, that you were using the archive, because that's been a pet project of Sarah's for a long time to get that together. And it's always rewarding to have someone actually utilize that stuff. Fred is curious to know, is acceptance of bioengineered chestnut within the scope of your studies? And if so, any insight into views in the US versus Italy, or US and Italy?
Yes, I'm looking at that. My research advisor kind of nudged me away because I'm trying to make this a 25-page paper, and I've already gone down several rabbit holes about bioengineering, biotech, and GMOs, and the perceptions of those, especially in relation to American perception of selective breeding and eugenics from the early 20th century to how that's gone to the wayside, how sometimes that has a negative connotation lasting to today, how some people think that GMOs are always bad, or selectively breeding chestnuts for characteristics that we desire can relate to that. And my advisor suggested that that can be an aspect and should definitely be a consideration when talking about the ethics. But in terms of my research question, it doesn't touch that as much, the comparison between the countries.
Yeah, I feel like I'm sure there's a lot that you can do between the two. Great. Well, we have under a minute left, and we have a rather interesting question from Cynthia. She says, I love what you were doing. I wonder if you noted different philosophies of beauty, also religious attitudes toward responsibility for creation.
Oh, that's interesting. I
don't know if you can get to all that in 30 seconds, but
yeah.
I'll let you try.
Beauty, yes. I've definitely seen ideas of what chestnuts should be. Sativa and dentata look similar, but there are definitely differentiating characteristics, and people want different things from their chestnuts. They should look certain ways. Religiously, I don't know. I'll have to look into that. Thank you for your questions.
Awesome. Well, hey, that was perfect. My timer just went off. So Jeff, thank you. Appreciate your time. That was a really interesting presentation and a topic that I feel like we don't tend to get as often within our sort of little chestnut group here. So thank you for your work and for utilizing Sarah's archive. Our last presenter is Maya. So Maya, you got 15 minutes, 10 minutes to chat, 5 minutes for questions, and that should take us right to the top of the hour.
All right, that sounds good.
Kendra, actually, before Maya goes, if I might add, so for our presenters, Jeffrey, Uma, Sky, and Tyler, there's some questions in the Q&A for you guys. It looks like we're probably not going to get to those by the end of the session. So if you guys don't mind typing in answers, that would be you are not obligated, but would be marvelous if you could type in the answers to the questions that appear directed at you. Sorry, Maya. Thank you.
All right. Is everything working right?
Yep, we can see your presentation.
Okay, great. So my name is Maya Niskutch. I'm a new master's student at SUNY ESF. I'm working mainly under Dr. Newhouse. And me being new, my presentation is going to be more about what I plan to do rather than results, but I hope you all still find that interesting. So my research is going to be on understanding the natural pollen dispersal of American chestnuts, as well as the effect of controlled burns. And so you can see that this is a pretty, I have two pretty separate things going on here, but they are related. The way I see it is they both will contribute to restoration of American chestnuts. They both inform, best practices in restoration and management. And this photo up here on top with the people conducting the controlled burn, that is actually of a controlled burn that I witnessed, that where we did burn some chestnuts. So I will get into that. It's not as scary as it sounds, I promise. But yeah, I've got the two main aspects, the pollination distance and the seedling fire tolerance. So this is the research site where both of these experiments will be occurring. The plots outlined in the green and the orange are the controlled burn plots, and the little starburst I have there is the pollination distance experiment. So they are on the same site. However, all of the interesting timings of such as like burns and the pollination season, they will be completely separate times, so they will not be interfering. And this site is in Tully, New York. It's about half an hour south of Syracuse. So to get into the distance pollination experiments, we have a central plot of transgenic chestnuts and these radial spokes with the triangles on them that have chestnut hybrids that do not release pollen. And the effect of this is that the only source of pollen will be coming from that center plot. So we will be able to see after a pollination season how many fertilized burrs we're getting at each little triangle of trees, and be able to determine from that how far the pollen is traveling and effectively pollinating trees. And so we will also likely compare the pollen that is in the air to the pollen that is fertilizing burrs on these trees by placing pollen traps at the different triangles to compare directly what's in the air versus what is actually fertilizing trees. And we will also be able to see, depending on the fertilization, if it is impacted by the direction of the wind. And if it is impacted by the direction of the wind, maybe we've seen a lot of the literature coming out recently that is contributing to this debate on whether chestnuts are insect or wind pollinated, and looking at how the pollination experiment goes, we could be able to tie that to whether or not the fertilization is affected by the direction of the wind, which may add to that evolving literature. And for the controlled fire, what we've done is we are collaborating with another professor, Dr. Vandriot, who is new to ESF, and he's doing these controlled burns for a class, actually, for a classroom setting, to educate students on how to conduct controlled burns effectively and safely. And so what we've done is we've planted American chestnut seedlings, very small, only like 6 inches high or so, into these plots, and we're gonna burn them and see what happens. But why are we doing this? Why are we burning chestnuts? First off, the seedlings that we are burning, they're not transgenic, they're just wild type, and there was no other plans for these seedlings. And we are burning them for a purpose as well. There's a lot of anecdotal evidence that chestnuts are tolerant to fire. And if they are tolerant to fire, this would mean that we could use fire as a tool to help establish them in restoration efforts. But when we burn, the chestnuts would survive, and the trees and plants that aren't tolerant we could, kind of hold them back a bit. And that would release the chestnuts from competition and help establish them better compared to, competitive trees that might overgrow them, like red maple. And some of the things that make us think chestnuts are tolerant to fire are just some of the physical traits they have, their vigorous resprouting capability. We see that a lot in other fire tolerant species. That if it burns to the ground, it'll have the energy to just grow back nice and strong. And also a lot of anecdotal evidence. Some of you may have seen this yourself, that wildfire comes through and then you come out a season or two later and the chestnuts are just doing really well, or at the very least they're surviving where a lot of other things didn't survive. And so back to the experiment, we've got some control plots, to compare, but there's also some plots that will be burned in fall, some plots that will be burned in spring, 6 of each, and each of these 6 plots will be burned with a different method. And these different methods are the image you can see on the right. They kind of show you how we're lighting the fire, and that affects the intensity of the fire in order, to get the best practice. How strong of a fire do we want it to be? How hot? How fast? And we should, hopefully get some idea of that by, burning with all these different methods that do produce the different fires. And like I said, we are not, the burns are not traveling anywhere near the trees that we are using for the pollination experiment. And it's just not at the same time, so they should not be affecting each other. And we did plant and burn some chestnuts already. We took measurements of all the seedlings we planted beforehand, of their root collar diameter, basically how thick the stem is at the base, how tall they are, how many shoots they have. But we are not measuring the results of this first burn until the spring to give the chestnuts a time to actually grow and respond to the fire. So there's not really any results for that, but we are, making progress on this. And so that is really most of my, that's my presentation. So if people have questions, I would love to answer them.
All right, Uma, or sorry, Maya. Great, looking at different things when I'm trying to speak. Well, so far we just have a positive note that this is a very important research. Good luck and thanks.
Thank you.
It seems like there's a little lag for folks to start typing, so we'll give them a minute. While folks are waiting, just a reminder, I'm going to put it in the chat one more time, but the poster session does immediately follow our flash talks here this afternoon. You can access that by going to the link in the chat, and Maya, Uma, and Tyler all have posters up there, and they will be monitoring the comments for about an hour, so that if you have questions specifically about their posters or follow-ups about their research, that's another way that you can interact with those folks. Cynthia asks, can you tell us anything about the history of using fire? I have heard it was a Native American technique. Any idea how they did it?
I have heard that as well. However, I don't really have more knowledge beyond that. I don't really want to speculate about something I don't know about. So I can say I've heard of that as well, but I don't really know how they did that.
Fair. I don't either. Any other questions, folks?
Kendra, it's Hill Craddock here. Can you hear me?
We can.
I'm interrupting because apparently Uma is a panelist. We can't ask questions in the chat. So
yes, that is actually the case. So go
I'm
ahead.
just going to barge in and ask Maya. Maya, very good talk, by the way. I enjoyed hearing it. Maybe I missed this and you said it. You showed pictures of burning. Did you burn the seedlings already?
Yes, we have burned the fall planted seedlings. So yes, we have burned trees already.
So excuse me for being a little bit distracted. How do they look now? Like, can you tell? Did they burn off to the ground? Did they stand up? Are they gone? Are they?
Yeah, definitely depended on the tree. So the burn itself it was a bit wet that day. It's pretty hard to burn in New York in the fall in general, but so the burns themselves weren't very strong. They definitely, the plots did burn, but they weren't, they weren't very big burns. But it depended on the tree. There were little seedlings, and they did try and like purposely go in and like pour the gas right on the trees. When they did that, they like, poured the fire right on the trees, they burned down to the ground. Some of them just had singed leaves. It really depended. I think probably just because, it wasn't a very good fire. But they did go in and pour gas right on some of them where they could. So some of them were just empty sticks. Some of them were burned to the ground.
Were you on the fire team?
I was not on the fire team, but I did observe that day.
Did you direct them to pour the burning gasoline on your trees?
Well, I guess it was more next to the trees. But we did kind of have to do that to get any fire near the chestnuts.
Okay, all right.
Yeah.
Interesting. So you'll be measuring the survival in the spring when they leaf out or not.
Yes, exactly.
And when is the spring burn scheduled? February, March? When do you burn in New York?
I think it's March or April.
Oh, okay.
But I'm not too sure, and it does depend too, just on conditions. But I think it's March or April. That should be a better burn. Spring burns are better, apparently.
Awesome. All right, we do have a question from Matthew. He wants to know if you have any guess on what the pollination distance may be.
Not really. I feel like there's, anecdotes of, trees in the middle of nowhere getting pollinated. But I mean, I really don't know. Yeah.
All right. Well, I think it'll be interesting. I think that's going to be some really interesting documentation, just because I don't know that we have that. So,
yeah,
it'll be pretty fun to track. Let's see. We have 2 minutes. I'm not seeing another specific question for Maya. So I think I'm going to say thank you all for coming. I'm going to say again, please go check out the student posters that are posted on the website. It would be really great to have some participation in our virtual poster sessions. The first time we've tried doing a virtual poster session. Certainly open to feedback on how that goes, either as a presenter or as a observer or participant in the virtual poster session. And thank you again to all our students for putting yourselves on the hot seat and sharing your research and getting grilled by all of our chestnut enthusiasts. We really appreciate your time and effort. And I think that's it. Sarah, you got any closing thoughts?
I do not. Other than thank you. This was awesome. Really enjoyed it. Loved having all like the diversity of projects and people and students here. So thank you guys for participating. November 17th, Michael French, Green Forest Work, Mineland Reclamation, come next month. But before that, switch over to the link so we can continue the live poster session. Thanks, everybody.
All right. Thanks all.